I'll try to put it in lay terms. And as Bob described it, we at UC Davis, well, a number of universities in this country, in Canada, other places of the world, have been working for quite a bit on developing tools and new technologies that were quite cool, interesting, and useless. So, and that's pretty much what Bob said when they hired me. So the reason why I appreciate that, and that's actually the reason why I accepted to move here and be more in contact with the real world and the applications that we can actually have to help people working out there. So with that in mind, I'd like to introduce some advances of this work in progress. So we are just starting to develop this analysis. Hopefully, we'll have some results on this soon, but I think it's still interesting as we move forward to get some feedback and to get people aware of what we are doing. By no means this is my work alone. This is a teamwork. This is a team group work, and a number of people that have been involved on that. I'm just the presenter here. So I'd like to start by acknowledging Mo Alkamis, Mike Mortag, and his students Xiang Wang and Bob, who are the leading forces here, and some 4 anonymous participants of the Swine Health Monitoring Program that helped us to develop these models and to search for these applications. So let's start with some basic introductions to make sure we are on the same page. What I'm trying to do here is to show some basics of molecular epidemiology and why we are interested in that and those tools and how these tools may help us. So each of the blocks here simulates what may be a virus sequence. And each of the squares would be what we refer to as a site, or you may look at those as nucleotides. So the variations in those sites and those nucleotides will result in encoding. Each of those sites will encode for different sites in the virus. And variations there, which are quite often, will result in differences in the amino acids of the viruses. And then those differences in the amino acids of the viruses will result in different behavior of the viruses in the transmission. Some of them will be more transmissible, some of them will cause more disease than others. And all of that information is in the genome of the virus. That's where all this information is seated. So if we can learn more about that information and that relation with the clinical presentation of the disease, we may be able to get a little bit better in our predictions. So suppose that we have sequences from 8 farms. 1 sequence per farm. Each of these blocks represent that. Each square represents 1 nucleotide of the sequence. The first thing that we do with this information, when you submit your samples to the lab, the samples are analyzed, viruses are identified, then people are way smarter than me will sequence those and will identify the sequence, and they will tell us what are the nucleotides for each of those sequences or each of those viruses that were on each position. So now it's like having a sort of fingerprint. We have a map of the virus that was causing that outbreak. And the first thing that we do is a process that is called alignment. We align those sequences, and now we can compare the genetic composition of each of those viruses, the viruses that were in your farms. And after we can, and what we are doing then is aligning there, and you can see that the first, for example, the first position, the second position, this is simplification. There are hundreds, thousands, depending on the virus, of nucleotides on these positions. So this is a simplification. But now we can compare. And you can see each color here represents a different nucleotides. There are 4 possible base pairs that may be found here. So there are 4 colors here. And then we can compare and see in which positions they are similar and in which positions they are different. Okay? After we have done that, we can construct the matrix. And with that matrix, we can do pairwise comparisons of the viruses that were found in the systems in the farms. So now we can, comparing those, the structure of those viruses, we can say, well, farm 1 compared to farm 2, there were 2 nucleotide difference in those farms. Other farms, they may have 4, 3, 7, whatever. Then with that information, once you have the number of different nucleotides, if you divide, just to simplify it, if you divide that by the number of nucleotides that you have analyzed, you get an estimate of of the likelihood of those viruses being different or similar to each other. at the end of the day, it's a probability of being different. So if the differences are 0, that means that both sequences were exactly the same, were identical. If there is one nucleotide difference divided the total number of nucleotides, that gives me a probability of those one side being different through that sequence. So basically, what this matrix will tell me is the probability of finding one different nucleotide when two strains are being compared in this matrix. So now with this information, I can estimate how similar the strains are compared to each other, how different they are. With that information, what we do is we build dendrograms. These shapes here are called dendrograms, and if they are done in the context of phylogenetic analysis, they are called phylogenetic trees. And what we do there is try to establish those relations. So we take the information in that matrix and then put that information together and then try to build a tree to see how the virus may have spread and moved and changed between the different farms and the different systems. A little bit of nomenclature here. We call topology to any potential pattern of the tree. And taxa is any kind of taxonomic unit. So for example, in our analysis, the taxonomic unit that we are analyzing are the virus sequences. The problem that we have is that there are many potential shapes, many potential topologies for any combination of taxa being analyzed. So if you have 4 taxa, only 4 viruses, only 4 farms being compared to each other, there are 15 potentially rooted tree topologies that may be the true, which is we don't know. What we don't know, that's what we are trying to elucidate. Those topologies are showing how the virus have been bifurcating. So maybe that there was an origin of a virus, and then it bifurcated in farm A and B, and then from there to farm separately to farm C and D. Or it may be that it started here and then bifurcated in farms A and C, and then B and D, and so on and so forth. There are, for 4 viruses, there are 15 shapes, 15 topologies in which this virus may have spread in the population. Okay? And only one of those is true. That's the problem that we have. When 10 taxa, when 10 viruses, suppose that we have 1 virus per farm, when 10 farms, 10 viruses, 10 outbreaks are being compared, the number of possible rooted topologies is 34 million. Okay? And only one of those is true. That's the problem that we are facing. That's why we try to estimate. We never know the truth. We try to estimate what's the most likely relation of those viruses, try to infer what was the most likely way of spread. And for doing that, we use different methods. Some of them are traditional methods that ignore the association of epidemiological information, such as space, time of identification, or associated data. And some of them will try to incorporate that information into analysis. So let's take a look. Let's try to take a look at this with an example that's completely unrelated, but hopefully I think that it would make sense. So we are all familiar with this story, right? What I didn't know before I read this paper is that actually the same story has been told on the 5 continents in different periods of time. So the story was there. We tend to believe that it's coming from Europe, right? And then we received it from our ancestors that migrated here and then told the story. But what's the true origin of this story? And it has been told. We told it to our sons. Our fathers told us to us, and then it goes down and back and back and back. Who knows until where? And the same story has been told in the 5 continents. So what's the true origin of this story? Same situation with the viruses. We are seeing viruses in different locations popping up and then transmitting and moving in our systems in the farms. But what's the true origin of those viruses? How it evolved? So what these people did is that they built this dendrogram, which is a relation of the way in which the story was told. So in some cultures, the story, there was a little girl, in another culture was a little boy, in some parts of the world it was red, and in a different place it may have been blue, and they combine all that information along with the origin of the story, the place of origin, and the time it was first described there, and then they group the stories. And with that information, they were able to follow and trace back the movement of the story through time and through space. And this way above my head, but I believe that the conclusion is that they found that the origin was somewhere in Asia or Africa. And then from there, it moved through traders and spread into different continents, and the story is shaped in a different way until it took the form that we know now. So this is an analogy of what we are trying to do with these viruses. What we are trying to do is take the same information, what we know, locations, times in which we saw the outbreak, clinical signs, relations in terms of the systems, and try to put all the information together to try to see if we can estimate what's the most likely way in which the virus has been spread. Does that make sense? Okay. And in the process, we learn a few things. One of the things we learn, for example, is that initially we tend to we used to look at the differences in the viruses, in the PRRS viruses, only at the number of different nucleotides that we saw in the sequences. I have described before. What we learned in that process is that that is not an accurate way of differentiating PRRS viruses. So this graphic is showing here is that in the x-axis, it has the number of nucleotide differences that are in any pairs of viruses being compared. And then in the y-axis here is showing an estimate of the phylogenetic distance. So when I consider all the structure of the population, different in the nucleotides in that population, time of isolation, place of isolation, what's the most likely phylogenetic distance in terms of evolution? So it's trying to show that correlation, and at first sight you may think that it's a good correlation, but then you find things like this. So viruses, when you compare the pairs of viruses, so two viruses, for which the number of different nucleotides is quite similar, it's in a given range, but then the phylogenetic distance, this is the history of evolution, is quite different. So they may look the same, may look similar, but actually the way in which they evolve is completely different. Maybe in two different evolutions coming separately, and then now look the same, but actually they were different. And then you have things like this in which you have a relatively recent history of evolution with quite a bit of variation, of genetic variation in terms of difference of nucleotides. And that's because the PRRS virus mutates a lot. So maybe related to each other, but they are changing and mutating a lot. Okay? So, similarly, what we can do, similarly to what I told before, what we can do is to use phylogenetic methods to infer the most likely time of emergence of the strain, the past probability of spread between systems and between types of farms, and the population viral size. And we did that with a sample of 5 independent systems. We collected the data that these systems and the sequences, locations, and time in which the sequences were obtained, the samples were obtained from where the sequences were obtained, between 98 and 2015. And those were more than almost 7,000 or 5 sequences. And then we ran a basic algorithm. We built a dendrogram to identify those that would be included or considered 174 PRRS viruses. And this is what we found. We found that almost 300 sequences were related to each other and may be considered part of this big family of relation collected between September 2003 and March 2015. And most of them, based on the sequences and based on the location, may be considered 174, obtained between January 2014 and March 2015. So, among all this pool of samples that we are collecting and that we receive from these systems, we screen out those that were not related to 174. And then we worked only with those that were considered 174. And this is not related with the RFLP. One of the things that we learned in the process, we knew that before, but we checked that here, is that just looking at the RFLP doesn't provide a good estimate of the relatedness of the viruses. So we still call them 174, but it's not actually based on the RFLP, it's based on the genetic composition. And then we analyzed that, consider the genetic variation, time of isolation, and place of isolation. And one of the interesting things we found, this graph here is showing the virus diversity through time. And in general, virus diversity, so virus diversity is the difference in the genetic composition of the viruses. In general, that virus diversity has been shown to be a good proxy for size of the virus population. So if you see that the viral population is changing, is different, more different now compared to what was in the past, that may give you an idea that the size of the virus population increased. Because otherwise you don't get that wide range in variation. So what's showing is that sometime in 2013 there was an expansion in the virus, strictly speaking, in the virus composition. So in the variation of the virus, the genetic diversity of the virus. That's exactly what it means. But we may think about this as a proxy for virus population size. So the number of viruses of these families that were out there. So this model, it's a model, seems to estimate that the increase occurs somewhere here, and then it resulted in the cases that we saw shortly after that. Now, another thing we can do with this information is to try to estimate the history of a spread between systems. And again, this is getting back to Bob's point and Paul's presentation earlier, the value of getting access and sharing information and being able to analyze that. This may be biased by the number of systems in which we are working. This is a pilot study with only 5 systems. We believe that if we get more information and more systems in, we will be able to be more accurate. But just for a moment, let's focus on these 5 systems, and let's assume that this is representative of the population. What this type of models may help us to see, and what we're showing here, is that the colors represent each of the systems. So what we see here is that during some time, 2002, 2007, this family of viruses were sitting in this system. And they may have been hitting this system for quite a bit. And then at some point something happened, and it spread and affected their systems. But actually, this is system C. But actually, seems to be system E being the one that is connected to all the others. So the virus was quite quiet here, in a sense. And then there was something that may have changed in the dynamics of the virus, and that may have been related with this system or not. It may be that this system just was there and was the one that was hit by the virus. But then something happened here, and from there, they spread to all the other systems. The other thing we can do is just, rather than looking at the system as a discrete variable of analysis, we can look at the type of farms, and then we can compare that in terms of evolution and change. So what we're showing here is that basically it was sitting in sow farms, and then from sow farms spread to other type of farms, which is not really surprising, but it's always good to see. And when we find this type of results, we believe more in the things that we are speculating with other things. Because if it makes sense with that, biologically speaking, then perhaps other interpretations that we are doing also may make sense. The dynamic representation of what the things we can do in terms of the spread. So the area here is showing how confident we are that the virus was in that system. Each of the dots here represent one of the systems. So these are not the real locations of the farms, just representing locations of the systems. And this is not spatially structured. So in order to protect confidentiality, we just put these dots in Alaska, somewhere. And that's not even related to the relation that these systems have. They are just for geographical display. But the circle, the area there, is showing how confident we are that the virus was there in that particular system. So we are confident at the beginning of the study that the virus was sitting in this system. And then, if I can make it work, it's showing how confident we are that it's there, and it's showing the most likely spread between and among the systems, and how confident we are that the virus is there. So you can see that the expansion that goes into system E, when we are confident that the virus is actually entering there and spreading from there into our systems. Again, this is work in progress. We are not jumping into any conclusion. We are working with a biased sample because it's just one system that voluntarily shared information with us, but shows the potential of the things that we can do if we were to work on this on a routine basis and being prepared when this happens to do it in near real time. The conclusions. The conclusions, again, is that this is work in progress. And what are the next steps? Next steps is to keep enrolling new participants and new people. That's critical. It's critical to increase the accuracy of what we are doing. Keep learning and see what are the applications of what we learn out there.